So it's not just psychiatry or oncology, cardiology. There's been limited uptake because alternate agents have come along, but it is a recommendation, and it's a black box recommendation by the FDA. So for example, if you're a cardiologist, you use clopidogrel, they're a poor metaboliser, they have a clot, they die, their wife sues, they find on post-mortem they're a poor metaboliser, they got an attorney. This is already happening in the USA, not yet in Australia. I'll skip that. Carbamazepine causes a skin condition called Stevens-Johnson syndrome, which is also seen in other aromatic anticonvulsants like lamotrigine, phenytoin, other agents like allopurinol. This is sort of an extreme immunological allergic reaction to agents that can be so bad it's like third-degree burns. Toxic epidermal necrosis with death happens. Not for the faint-hearted, but we need to look at this slide, which is Googleable. That's a child tried on an anticonvulsant. And that was the adverse drug reaction. So sometimes that's what an adverse drug reaction looks like. Can be quite fatal. There's a fluid loss. So this is one of the areas which I think we need to know today. And I think there's hazard if you don't think about it today, which is HLA genotyping for carbamazepine, maybe lamotrigine, but for carbamazepine, you don't have to know which variants of the HLA immunological genomics is relevant. You just need to flag with a patient, particularly if they're of Asian ancestry, that if you need an agent associated with Stevens-Johnson syndrome and carbamazepine's got a depth of association data, these rare catastrophic adverse drug reactions will not lend themselves to being studied through randomised controlled trials for two reasons: power, large N needed, and ethics committees who won't want to necessarily let people have hazard in one group. So large association data sets are sufficient data for these catastrophic rare outcomes. So don't wait for RCTs. And it's already in for this. So there's a 348-fold risk in certain Asian populations. This one, a group from Thailand. These have been replicated many times. So this is now neurology guidelines. If you're using carbamazepine, you should offer people, whether they take it or not, the genetic test on HLA, especially if they've got an Asian background. Because if they get that fatal reaction, it's on your head. So there's multiple legal cases in the US. $75. No, no. Yeah, there's a lot of things like that. Yes.
I had two patients with very small doses of lamotrigine, Han Chinese, both of them, and there's a big study now in China, specifically HLA related as well for lamotrigine.
Yeah.
Two patients, very small doses, just came up with a rash, and with elevated liver enzymes.
Yeah, and wouldn't it be fascinating if this stuff one day is shown to be another biomarker for people at risk of autoimmune, neuropsychiatric presentations? You could even go back and start doing it. $75. It's cheap.
So what do? You fill out a Melbourne Path form, or you have to go to a particular lab, and what do you write exactly?
I'll show you later. But it's simple. But we do need to do it now.
It says with Caucasian patients as well.
So it's relative risk. So I think that most of the neurology guidelines globally say in Asian patients it should be offered. But people are Googling this now, and so they might say, look, I'm going to go on, I hear carbamazepine is sometimes used in bipolar, you suggest it, or you don't suggest it because many people don't use it anymore, you suggest lamotrigine, and it's a cousin of carbamazepine or in the same group. And so I think being able to give them an answer saying, oh look, if you're a Caucasian background, the risk is low, but it's $75, so let you decide. A bit like meds in pregnancy, it's category B, C, but let's have a dialogue about this. And I'd manage it with the same clinical skills you use in those situations. My view is for carbamazepine, definitely. Lamotrigine, on the cusp of global consensus from replicated association studies. Or you can just do the really super slow titration, which we do across the board. So I mean, an interesting another way of looking at it is, And the flip side, which will be trickier to implement, is say you've got a genotype that means you're low risk of Stevens-Johnson syndrome, and you can whack them on 200 of lamotrigine day one. Now, that will be maybe more clinically relevant in a way, because at the moment we just do global baby dose build-up. Designing those studies, getting ethics approval will be a challenge, in fact probably impossible. So also when you look at the literature, if you notice glaring deficiencies, sometimes it's because of ethical issues, and so there'd be difficulty getting an ethics committee to accept one limb of your studies is going to be whacked on 200. It'd be very difficult.
Sorry, Ajeet, the HLA marker for lamotrigine, is it the same marker or a different marker?
There are more than one. So it's probably a haplotype block, but haplotype blocks are only probabilistic. So some studies say the block is this, others say it's this, others say it's that. So there's a bit of wiggle room. There are some, though, that have got strong association data, and I think over time the supercomputers will put a lot of data together and say your composite risk for Stevens-Johnson syndrome is 500-fold. And then we can use that in the clinical risk-benefit decision-making. Yes, at the back?
Sometimes it's very complicated. For example, I've got a family where you had Stevens-Johnson type reactions to, in the mother, galantamine, and in the daughter, carbamazepine. And I also want to mention that when Stephen Starr was speaking about desvenlafaxine when it first came out, one of the selling points was that it didn't interact. It didn't inhibit the metabolism of codeine into morphine, as did venlafaxine.
Yeah, desvenlafaxine is an interesting one because it doesn't have any phase 1 metabolism. 10% of people at phase 2 are poor metabolizers of a thing called UGT101, but that's 10%. But it's a big dose range clinically, and my view is that's because of blood-brain barrier genetic variability, which is my pet area. So HLA, carbamazepine, yes. Lamotrigine, probably soon. People being sued in the US, but when it goes to court, the expert witnesses say, carbamazepine, no, you need to. So with psychotropic pharmacogenetics, the questions we need to ask are, can we reduce trial and error prescribing? Is there an affordable, accessible test, which is what Kate asked before? Will this reduce time patients are unwell, improve their outcomes? Will it reduce cost of care? Can we predict adverse reactions? So any paper, don't be bamboozled by all the gobbledygook genomics. Think this is what I need to know as a clinician, and if it doesn't address that, toss the paper. That's what I'd say. Others would disagree. When we get back to the that CASPI study with the stratification of serotonin, there was hope that maybe in 2003 we got a biomarker for risk of suicide. It didn't pan out. I'll skip that one. When they did studies looking at serotonin transporter, they found a definite effect on response to antidepressants, for example, but the effect size was teensy. So like if you've got a test that increases your predictive power by 0.1%, who cares? So unfortunately, without any methylation studies, just looking at the genotype, there wasn't an effect size. Last year, the first methylation study of response to antidepressants looking at serotonin transporter was published, including with a local professor in Adelaide. They got a big effect size. I'm very excited. I tend to get excited. So they got a big difference of if this serotonin-linked transporter polymorphism, the short version, stratified by whether it's switched on or off, the methylation epigenetic element, they got a big difference in extent of improvement percentage-wise on the depression rating scale. So it may be that we'll revisit serotonin transporter as an odds of responding to an antidepressant. But I'd argue as a clinician, if we're going to try an antidepressant, we've already gone through conservative measures. So it may be less of a clinically salient question if we're at the point where we're going to use a medication anyway, whether somebody's 10 or 20% more likely to respond, would that sway our decision? So again, the clinical context is key. Methylation is, I think, going to come through. We'll see one study not replicated, but it's interesting. It fits all that science. That's why that science is now relevant. That's why I'd argue it's worth learning.
I wonder whether it's actually worth thinking about it from a patient perspective, though. Sometimes information to people who have difficulty accepting they need treatment may be a use for that. you know, like your risk is actually increased because you have this abnormality, this difference from other people, and, you know, they may invest more in treatment? Do you think?
Yeah, so I think, I mean, basically all the new technology is housed in old clinical wisdom of the doctor-patient relationship, having a dialogue, and trying to figure out what's the reasonable risk-benefit for anything, and then shaping that to the emotional state, educative level, degree of involvement of the patient. So I think a lot of what we do every day in our practice as clinicians is as relevant as ever. And I think the analogy is when you talk about risks of agents in pregnancy, and we're used to having those dialogues, and then when you get genomic information starting to influence those dialogues using those same sort of skills. Because we don't want to scare off people who might benefit from a pill, but there is a risk. So this sort of probabilistic type medicine is going to be increasingly relevant.
I wonder if there is any evidence, do we have any information about whether past or current use of any psychotropics actually affects the methylation and gene expression?
Yes. Beautiful question. So I actually think that it could be part of the mechanism of action of psychotropics, is it might help switch genes on that help recycle monoamines in the frontolimbic parts.
So the implications for research then you have to control your patient populations for having ever been or not been on particular psychotropics, etc., etc.
Yeah, so it's complicated. The other sort of complication with methylation is it's tissue specific. So when they look at methylation profiles from a peripheral sample, it's not necessarily a one-to-one correlate with what's happening in the brain. So I think you're right, and there'll be more questions than answers, but you're thinking correctly, one step ahead of what's going to happen when they try and look at epigenetics as predictors.
So the question that's relevant to my practice, I use genes effects a lot.
Mm-hmm.
The question is, should those tests be done prior to commencing patients on any treatment? And are they affected, are the results affected by the fact that the patient is already taking some medication?
Yes, a very good question. So good that I have a suite of slides to follow, because it's a key question. So there's an evidence will reach a level where you say you should do it before you're prescribing, and there'll be an evidence that say if you happen to have the data, what should you do with it?